As was mentioned, I study the microbes associated with guts and the foods that we put into our guts. Melissa and Andrea both mentioned microbiomes yesterday, and from what I understand, it's becoming more and more of a household buzzword. So I'm really excited to hopefully shed a little light on the way that microbes contribute to the good health of whole grains. So gut microbes actually contribute to good health across your lifetime. From birth, the gut microbes help your gut to actually develop. So when you are an infant, like my little girl upstairs, you have a very different gut from the guts that you have today. And gut microbes are actually, by digesting food and producing short-chain fatty acids, vitamins, all these different compounds from fiber and other nutrients that are unreachable by you as a host animal eating those foods, they're actually providing key nutrients that can help your gut to form and specialize across your lifetime. The gut microbes in forming those products, they not only affect the tissues in your gut to form and specialize, but they are also sending chemical signals to your brain. So gut microbes, increasingly, we find, affect the moods, behaviors, even the foods that you crave. Those aren't your cravings necessarily. Those are the little teeming trillions, right? Your superhero sidekicks. So they're really incredible partners in life. They also affect medical markers of good health. For example, of obesity, diabetes, and heart disease, there's increasing research that demonstrates that gut microbes are tied up in all of these different conditions and in the prevention of those conditions. And they do this by digesting their food and your food, right? So everything that you put into your mouth, into your gut that you can't digest, fiber, artificial sweeteners, all sorts of compounds, you better believe that those hundreds of trillions of microbes are going to figure out how to digest that since you can't. So everything you're eating, I'm eating for 2, but I'm also eating for 100 trillion, and so are you. So I've been really, really pleased with the amazing spreads that we've seen so far because they're not only delicious and highlighting whole grains, they're also full of excellent nutrients for your microbiome. So beyond the individual lifetime, microbial cultures lie at the heart of human cultures. We humans have been baking bread for over 10,000 years, and before refrigerators or commercial yeasts existed, we fermented foods to improve shelf life and nutrition. I was really excited to start my postdoc because I can bring my work home. I can experiment with it in my own kitchen in ways, and talk about it at meal times in ways that discussions about fecal samples and gut don't really translate. So I'm excited for the palatability of my new research systems, but I'm also really excited and invested in the human cultural history that I'm able to tap into. So when we ferment foods like this, the bacteria are improving shelf life, but also nutrition, again, because they're doing a bit of what gut microbes do inside our bodies before we ever even put these foods into our bodies. So you lose a little bit of what you put into your body, right, 8 to 12 hours later. Euphemize that a little bit, right? We don't always get everything that's going on, all of the benefits from our microbiomes before they're voided and gone forever. But if you ferment food ahead of time, you can actually get a little bit more out of it because those microbes have already started to perform these beneficial processes before you get to tap in and digest. But until very recently, no one had actually researched these microbes that contribute to our food traditions. So the Dunn Lab is addressing that gap with the Sourdough Project, a global citizen science initiative. What we did was to send out a call to the general public. So citizen science, very broadly, is the engagement of the general public. You don't have to have formal training or lab equipment necessarily to engage in citizen science. And we actually recruited 560 participants who sent us samples of their own sourdough starters from 17 countries around the world. So this is the largest library, the most diverse library of sourdough starters ever collected. And they're collected not from commercial purveyors or bakers, but from folks who do it themselves at home every day, the way that bread was traditionally made. So once we had those starters in hand, we used DNA sequencing to identify the bacteria and yeast in each sample, to see whether where you live or how you live determines which microbes live in your starter. So we discovered much greater diversity than was previously known to exist in sourdough. Here you can see a comparison of two starters, number 867 and 862. Those are their numbers as they responded to the survey that we sent out. How do you make your starter? How long have you been making a starter? Why do you love sourdough starters and baking? So they have higher numbers than the number of samples we have in hand, right? So you can see these starters are quite different from one another. The one on the left is very typical to what we would consider was known about sourdough starters before we started this project. So you can see the yeast present in starter 867. We have two strains of Saccharomyces cerevisiae. That's any packet of rapid rise yeast. That's going to be Saccharomyces cerevisiae. So this is a very typical starter from the yeast side of things. And that bacterially, it's also typical of a sourdough. It has Lactobacillus sanfranciscensis, which is from San Francisco, or is it? San Francisco's sourdough claims might be a little bit under threat from the microbial results that we're finding, though, their sourdough bread still does have a huge claim for being very iconic, right? So we're finding really interesting results here. So as far as the drivers of this microbial diversity. So we see a very typical what we thought sourdoughs were like on the left. And on the right, we have Kazakhstania unispora, a yeast that had not previously been identified in a sourdough starter. And the bacteria, Lactobacillus parliamentaris, to me sounds very British, but it's also hadn't been described in a sourdough starter before. And the light blue above that purple bar is Lactobacillus brevis, which is actually a plant associate. So that's starting to suggest that maybe the flowers and the grains are actually contributing to these sourdough starters. So we see a lot of diversity here. The surprising thing, perhaps, is that these starters, one is from Raleigh, one is from Durham. So they're from less than 30 miles away, and they are incredibly diverse and unique. And when we looked globally across our data sets, we found that broadly the yeast follow geographic distributions. Where you live tends to correlate with what yeasts are present in your sourdough starters, whereas bacteria follow in-house rules. So what type of flour you might use, how many times a week you feed your starter, if you keep it on the countertop or in the fridge, if there are males or females living in your house and working with the starter, and how many people are in your household. So all of these in-house factors tend to drive bacterial diversity. So that was pretty interesting to think about. But where are those microbes actually coming from? Each of these sourdough starters is made from flour and water, grown over time, and even the flour is a product of grain. So there are several steps that occur before we actually got these snapshots of mature sourdough communities. So in order to kind of investigate how these earlier factors might contribute, I developed a citizen science lesson plan or two to engage public school students in authentic research experience to study the microbes associated with flours made from different grains. So first, at the North Carolina Governor's School, I had 66 gifted students come in from natural science. And when I sent them their welcome letters, I said, I'm excited to explore science with you. Make sure you bring a quarter cup of flour from home. Having no idea what they were going to get into, they showed up, they're like, I brought my flour. And you can see a small graphic of the geographic distribution, which was incredible diversity for our program at the Governor's School. And then also from that Ziploc bag, we then mixed flour and water in tubes, they shook it, and then we took some of the supernatant and plated it on separate Petri plates to grow up microbial cultures of bacteria or yeast. So while we waited for those cultures to grow, because it takes about 4 days, we actually kind of backfilled information by studying the grains that those flours were coming from so that the students could develop hypotheses. So I'm preaching to the choir here for sure, but you all know if you look at a grain, you have these different components of grain, right? So this is almost my test. How accurate am I teaching the kids? We have bran, which tends to be a source of a lot of fiber. Endosperm is a starch bomb. And then we have germ, which is going to supply some protein, right? But what a lot of people might not be thinking about when they pick up a grain, or a bag of flour, is that the bran and the germ also supply microbes. So the bran tends to have environmental microbes that may even come in from the fields where that grain was grown, while the germ has very special specific microbes that are in such a tight symbiotic partnership with the grain that they're allowed to grow inside the germ of that plant. So we have, for different flours made from different grains, not only do we have different nutritional components, right? So our all-purpose white flour is going to have a lot of starch, whereas whole wheat not only has fiber, protein, and starch, but it also has those two types of different microbes sourced in with the grain components. And rye also has those three components, but in a different balance. So rye tends to be more starch heavy. So these different flours have different nutrients, but also different microbial inputs as well. So here's what we found. White flour, I'll show you a picture of a Petri plate with the bacteria on the top half for each of these flour types and yeast on the bottom half. So white flour tends to contain a lot of lactic acid bacteria, but no yeast, right? So we can think about those microbial inputs that might have not made it into that starch-heavy flour. Maybe those environmental and germ-centric microbial inputs are actually yeasts that are being omitted from our all-purpose white flour. Whereas whole wheat flour was really surprising. We had an opposite effect. Whole wheat had no lactic acid bacteria growth, but there were a few different types of yeast. So you can see that one larger colony toward the center with the green middle, right? That's a different type of yeast than all the other really shiny little kind of snot droplets, right? I'm not gearing you up for lunch so much, am I? And then rye was another surprise. Rye had a mixture of diverse lactic acid bacteria as well as a few different types of yeast. So again, we see differences not only in nutritional input, but also microbial input. And these differences are important because microbes contribute to the flavors of bread. We have bacteria digesting the starch to produce acid, and acid is the sour flavor that you taste in that tangy sourdough, while yeast tends to produce the complex aromas that we smell and taste. So these microbial inputs and the differences in the nutrients in flour are working together to contribute to specific bread aesthetics and attributes that you might experience, for all you foodies who share my love of bread. So it looks like flour affects the microbes that change the flavor of bread, but what about the growth and activity of the sourdough starter itself? When I was starting my own starters from scratch, I read anecdotally tons of blogs. Everybody says, add some rye flour, it changes your starter's activity completely. Has anybody else seen this? Yeah, okay. So we're reading some of the same blogs or sharing some of the same anecdotes. So I had a hunch that this is due to these differences in flours and microbial inputs. So to explore those impacts of the whole grain flours over time, I developed Sourdough for Science. It's a classroom activity that enables and empowers students to study microbial metabolism. And importantly, they can do this without needing a fancy lab setup. So I call this countertop science, because you can do this at school, on a classroom desk or a tabletop. You can also do it at home on your counter. I do it all the time. My husband is very patient. So with this activity, we ask students to grow sourdough starters using different types of flour and to measure the height, so the growth over time, the number of bubbles formed in that 1 centimeter square that the students draw on the jar, and the acidity every day with pH strips. And that helps to track microbial growth and metabolic processes without needing DNA sequencing or Petri plates or a hood, right? Any of these fancy lab setups. So to date, I'm really excited that over 200 students in 2 counties in North Carolina so far have completed the lesson plan. And the student evaluations so far indicate that we are helping to address both science education by giving authentic research experience and actually concepts of food equity, because a student is now learning, oh, flour, water, and thyme, and I can make something that will nourish my body for, hopefully more completely and at lower cost than a lot of other foods that they might be after. So getting a peek at some of the preliminary results coming in from these schools, what do you see? Yeah, so they got taller. We see growth over time, right? Those blue lines are typically going up. Anything else?
pH stayed about the same.
Okay. pH looks like it's staying fairly stable. Okay. What I saw when I saw this was, one, that the growth is not stable. It takes a while for these starters to stabilize. If you've ever started your own starter from scratch, you'll notice like the day 2 or 3, they look amazing, but if you try and bake with them, your bread is going to be a rock, right? Because it does take a week or 2 for these starters, the communities, to build up and stabilize, right? So we do need some time. So we see that kind of instability reflected in the growth. The other thing that I saw was that the white and the rye looked like they had very similar pH patterns, which I again would attribute to the starch content of those flours and the growth of the lactic acid bacteria, which are digesting starch to produce acid, which would decrease pH over time. So we can start to take all these different pieces of the puzzle and bring into a big picture of understanding. So to visualize these differences, I set up a time lapse with 3 mature starters that were fed either all-purpose white flour, whole wheat, or rye flour. We combined equal weights of starter, flour, and water in 3 1,000 milliliter graduated cylinders to compare the growth dynamics over an entire day. So as the video plays, watch and see what differences you can spot. I did think about dividing the crowd up into 3 spots so that you can all root for one. But.
Come on, whole wheat. You can do it. Who said science isn't fun?
I mean, I love science, and yeah, it's fun every day. So, everybody saw that okay? Okay. So to recap the highlights, given the same amount of starting resources, all 3 starters reached similar heights, right? Though the white and the whole wheat did grow the most. But they grew at different rates. You saw the big boom and bust. And then they fell to different low tide marks, right? So also regarding those rates, the white fed sourdough starter tended to grow the fastest, but also crashed more quickly, and it crashed really hard, right? While the rye supported a slower but more sustained growth, right? So you all saw that. I'm not making this up. Okay. So in this way, I think that the microbial growth in these starters actually mirrors human metabolism. We even see kind of a sugar crash after we're eating the processed foods and after our microbes are eating those processed foods, right? So in fact, I would argue that gut microbes, the microbes in the starters and including our own gut microbes, may drive that sugar crash effect or amplify it, as well as the beneficial effects of whole grains. So a little blast from the past here. Back when I was studying gut microbes in lemurs, I compared the metabolic pathways used by microbiomes in lemurs that ate mostly leaf-based diets to lemurs that eat mostly fruit. right? So here's a diagram of all of the pathways available. You can see a lot of sugar pathways are up at the top in the light blue and the purple. Then we have starches down to fibers for the TCA cycle in purple moving down. We have the hot pink is vitamin production. The orange is amino acids. And then in the green, we have more plant-related secondary compounds. Okay, so that would be also including some of those tannins that were mentioned earlier. So this is all of the metabolic pathways that are available to all microbes potentially, right? But when I compared the metabolic pathways actually in use by the microbes that were living in the leaf-eating lemurs or the fruit-eating lemurs, I saw some pretty drastic differences. Notably, the microbes that are fed leaves tend to digest those plant compounds in green, and including phytase, right? So we have phytic acid up at the top left. And that's actually helping to unlock and enhance nutrients in the food, while the microbes digesting fruit tend to not only focus on sugars, but they also produced some more inflammatory compounds. And the fruit eaters tend to have looser stools than leaf eaters, right? So we actually see they're not sick animals, but they do have some distinct differences in gut production, right? So bringing us back all the way to the beginning, it looks like microbes are very important for promoting good health, but also that whole grains are promoting the beneficial microbial fermentation that brings us that good health.